Okay. Um, Thank you all for coming. And it's my pleasure to introduce you to Professor Oliver Trapp, who leads the research at the University of Heidelberg on biotic chemistry and the origins of life. His work focuses on understanding how simple inorganic molecules could have evolved into complex organic systems capable of cell organization and catalysis. And today he will speak on from molecules to life, the chemical evolution from non-living to living matter. And please join me in welcoming Professor Oliver Trapp.
Okay, so thank you very much, and also good morning from my side. And I'm very happy to be here and to present some of our recent research. And I would like to start immediately, and I start with this picture. So I'm going back to the very beginning of our early Earth, so approximately 4.5 billion years ago. And I think you are all most familiar that initially a proto-planetary disk has been formed. And of course, at this time, we had already very important compounds which are essential to develop our organic chemistry later. So I also have to say that this will be a very chemical talk, but I hope it's very interesting to see how these really simple molecules, like here, water, iron, reduced iron, hydrogen, carbon dioxide, hydrogen sulfide, and nitrogen, essentially evolved in the formation of very simple molecules. Of course, at the very beginning, there was another impactful event, the moon forming impact with the Theia, and essentially this almost destroyed again the young Earth. But maybe it was also very important to introduce again a lot of energy. So a lot of heat was produced, and this is also very important for the chemical processes to start them. For us, we are focusing, or initially we are focusing a lot on the catalysis with iron zero, so reduced iron. So this was really abundant, and you can imagine, and I will show this later, that essentially very simple building blocks have been formed during this time. So by the reaction of hydrogen and carbon dioxide, essential and small building blocks are formed. So this continued, but over the time, iron became more and more oxidized, but also the complexity increased in these organic molecules. So the first nucleosides were showing up. So this is what is assumed what happened during this time. And then essentially, and this is what you always can read from textbooks, that at a certain point, the biomolecular machinery has evolved. So enzymes, DNA, RNA, and so on. And from this, the last union common ancestor, Luca, was showing up somehow. So people assume that protocells have formed, and these protocells have evolved essentially in the formation of bacteria, archaea, and eukaryota. But I have to say, the big question mark is essentially the time between these small molecules and this really complex biomolecular machinery that we know nowadays, what happened during this time. And this is where we are focusing our research, and we are highly interested, and we want to understand how this evolved. Because you can also imagine that even if you have all these nice molecules, essentially this biomolecular machinery will not automatically evolve in this time. I also want to show you a little bit historic development in this field, and it's very interesting. So mankind essentially started very early, and it goes even back to Aristoteles, who coined the term genesis automatus. So this is really this, maybe origin of life. So they were thinking about it, how everything has developed, and they also already understood that maybe there was a kind of an evolution involved. Of course, there was still at this time no idea about a molecular evolution. So this came way later. But it's also interesting to mention that there were many research and publications already starting beginning of 1800. For example, Wöhler very excited to make urea as a molecule, or then later Strecker, who made amino acids available. Then in the mid of 1800, Louis Pasteur, who is well known also for pasteurization, for example, of milk and so on, but he discovered optical activity, so the handedness of molecules. And this is very, very important. And then later on, Alexandre Boudlerow, at this time, he still tasted essentially the molecules they were making, the compounds they were making. And he discovered that from formaldehyde, he can make sweet molecules, so sugar, essentially sugar. And this was moving on. So I also would like to mention Alexander Oparin, who was thinking really a lot about prebiotic evolution and also about the atmosphere, the composition of the atmosphere. And he also coined the term primordial soup, very beginning of 1900. And then, of course, it's going somehow faster. 1953, the discovery of DNA, which was elucidated by Watson, Crick, and I also have to mention Franklin. So she did really most of the work. We should mention her, but Watson and Crick, they received the Nobel Prize, as we all know. And then also the famous experiment at the same time by Miller and Urey, simulating essentially a primordial atmosphere. So they discovered that by introducing sparks to a reaction mixture, they can activate nitrogen, and they can make, for example, also amino acids. So this is what they discovered. And then later on, of course, Leslie Orgel, he realized that DNA and RNA are very important, and postulated this DNA and RNA world, and also the evolution involving essentially these kind of molecules. Also interesting, Manfred Eigen, as a theoretical physicist, he proposed that cell formation is very important, and he was also thinking a lot about autocatalysis. So he was thinking about autocatalysis in a way that it goes in circles, like a hypercycle, he's calling this. And also almost at the same time, early or late 1900, Günther von Kitrowski did the first replication of RNA and DNA. So using just short pieces of RNA DNA as a template, and was able to show that essentially this assembles and polymerizes to longer strains. Also, Eschenmoser at ETH did really fundamental studies and was also looking in mechanisms, and in particular also the chirality of these molecules. I would like to mention Kenzo Soai, because he discovered a very important reaction also related to chirality. It is called an asymmetric autocatalysis. So we recently elucidated the mechanism of this very important reaction, which is somehow a kind of a prototype reaction for autocatalysis in general. In 1998, Günter Wächtershäuser proposed that sulfur cluster and iron-nickel cluster are very important to maybe induce exactly the process which I was describing to make these small molecules. And I think it is almost a fun fact, and I would like to mention it, that he patented the origin or how to make life. It's very interesting. Okay, and I also would like to mention John Sutherland and also Jack Szostak, because they somehow did ignition in this field. They initiated an ignition in this field. So people started again looking into it, because nowadays we have totally different analytical tools and methods to look into these reactions and maybe to understand these reaction networks that might lead to the formation of life. And this is, I think, very, very important that they initiated essentially all these initiatives about origins of life. Okay, I would like to start with something how I came to this field. And it started with looking at the composition of meteorites. And I realized that meteorites, especially iron meteorites, they are composed of iron, so approximately 92, 93%, 2% nickel, 2% cobalt, and some traces of iridium. And I realized this is really, really interesting, because this composition is essentially the ideal composition of a very famous catalyst. A catalyst which can convert, for example, CO and hydrogen into hydrocarbons. This is the so-called Fischer-Tropsch process. And so I came up with the idea that what happens if a meteorite enters a very dense atmosphere? So what happens, it will just heat up. This is also what we see still in the night, yeah, meteorites entering our atmosphere, but in this case, they get oxidized. But under the conditions of the early Earth, we had more inert conditions, so CO2, hydrogen, nitrogen, of course, and so on. So the particles are not getting immediately oxidized, but they are highly catalytically active. And they are so catalytically active that they can even activate carbon dioxide, which is under normal Fischer-Tropsch conditions not possible. And so we investigated this reaction, and what we found is that under meteorite catalysis, we can obtain a lot of formaldehyde and acetaldehyde. Maybe I can take it here. So this is acetaldehyde, alcohols, and also alkanes. And these are very, very important building blocks to build up essentially all building blocks or larger, more complex building blocks, which are essential maybe for development of life. So now let me show you how we do this. So we take real meteorites, we dissolve them, and we impregnate materials like silica or other minerals, which were also present on the early Earth. And then we reduce it. And by this process, we are simulating exactly this process, that meteorites are entering our atmosphere, making nanoparticles, and we get highly active catalysts, which we then investigate here in high pressure autoclaves, because we can assume that the pressure of the atmosphere of the early Earth was quite high, so up to 50 bars, maybe in a mixture of CO2, hydrogen, and nitrogen. We do then measurements, we identify all these compounds, we quantify them, and this was quite exciting. So here you see again how these materials look like. So also on different substrates like silica or phosphorite, and montmorillonite, which is also an iron mineral. And when we do this, we can, of course, first of all, we can vary the reaction time, and we see a quite robust formation of so-called oxygenated products. And this is, I think, very important, because many people claim that a kind of a Fischer-Tropsch reaction took place, but Fischer-Tropsch always leads to completely hydrogenated carbon compounds. And they are not reactive at all. So it's really hard to reactivate them, to utilize them to make, for example, other building blocks. The most exciting plot is this one. So where we vary here the partial pressure of CO2 against the partial pressure of hydrogen. So we did many, many experiments. And we have here essentially two lines here, which you can see here. So this is always the line where we have the mixture. So starting from 45 bars of CO2 and no hydrogen. So of course there is no conversion at all initially, or very slow conversion, because the meteorites in the presence of water, they get, of course, oxidized. They produce some hydrogen. So this would be a stoichiometric reaction. But as soon as we get more and more hydrogen formation, we see exactly this very robust formation of these oxygenated products, and only at very, very high hydrogen pressures and excess of hydrogen, then we see more formation of alkanes, which are essentially inert. This is a very important result, and this result also tells you that essentially this formation of these oxygenated products is really robust. It's not a narrow window where it can happen, but under really broad and very general conditions. So based on this, we also determined the reaction rates, and we are extending over the last years more and more our reaction network. So this is here only a very small reaction here depicted, but now in the meantime we have a whole reaction network, and we can implement it here in a model of a planet or our Earth. So I call it always the onion shell model, because we have always these shells, and we have exchange between these shells, and we can do kinetic simulations with this reaction network and see in which concentrations all these compounds are built and formed. And this is how it looks like. And this is, I think, very, very interesting. And essentially, it shows us that there was maybe already a first tipping point of our Earth, because what we assume here in the model is that we have a meteorite flux of 100 tons per year. Let us assume we have only 5% of this material are iron meteorites. And now out of these 5%, catalytically active is 1%, 0.5%, 0.1%, and so on. So less and less active material. And when you do this simulation, then you see that you need a certain amount of these iron meteorites, of these nanoparticles, to achieve a conversion of CO2 into these organic molecules like formaldehyde, acetaldehyde, methanol, and also ethanol. And what you see here is that because this model considers all energy fluxes and so on, that if the flux of meteorites or catalytically active material is too low, then we are getting into this, yeah, I call it the negative hydrogen production regime, which means that less and less hydrogen is produced, which can less CO2 convert into these versatile organic compounds. And this is very interesting. So essentially, at a certain point, if this happens, the planet really dies, and CO2 cannot be converted anymore into organic material. Okay, so now you have seen we made a formaldehyde and acetaldehyde. So the first question was, what can we do with this? And I mentioned already that Alexander Butlerov in 1853 discovered that under basic conditions, formaldehyde is converted into sugar type molecules. So, and this starts here with the formation of glycolaldehyde, glyceraldehyde, and so on. And then we are getting more and more of these sugars. By the way, and I think you are more a community which is also more mathematically interested. What is really interesting, if you count here the molecules, you will realize that essentially this is according to the Fibonacci numbers. And this means this is like a natural function that a network of sugar molecules is really growing. And it is nice to get all these compounds, but it is really a high complexity. So we looked into it. We also determined some reaction rates, but it's really, really highly complex because the problem is all these compounds, they can also decompose again and form more and more complexity, which is really hard to trace in such a complex reaction network. But we did it, and we looked also into side reactions. So there are side reactions, for example, the formation of, it's a kind of a disproportionation. So if you have, for example, glycoaldehyde, it can form also acids and alcohols. This is the so-called Cannizzaro reaction. But there are also other important metabolites which are already showing up, like lactic acid from this sugar network. And I think this is very interesting, but as I said, it is very, very complex. So we did many investigations, and we also discovered that this reaction also works in the absence of water or any solvent. It can even work under absolutely anhydrous conditions. So we are doing, for example, ball milling of the solid, and you see under the catalysis at the surface of some minerals, you see here, for example, the formation of these sugar molecules in really high quantities. Now, there is a small problem. So most people are interested in ribose. So why are people interested in ribose? Because ribose is an important building block to form DNA and RNA. And this is really a challenge and a problem for this Formose reaction, because under the Formose reaction, really only very, very small amounts of ribose are formed. And ribose, I have to say, is also not stable at all. And this is really a problem. Okay, and this is very interesting. So I was thinking about it, and it's also interesting that there is a whole community of people doing DNA and RNA research in this field, because these are important molecules. And it also goes back already to 1915. So Emil Fischer here in Munich, he was essentially the first doing experiments using, for example, these nucleobases and fusing it together with sugar molecules like ribose. What he discovered, and this was somehow rediscovered by Orgel, that unfortunately not the 5-membered ring is formed, but the 6-membered ring. And this cannot polymerize. So this is a real problem. Under these thermodynamic conditions, this cannot happen. And this is for sure not a pathway how RNA, for example, and also DNA was formed. I already mentioned that John Sutherland was looking in a different pathway. So he started here with an interesting molecule. This is so-called amino oxazoline, oxazole. And he's fusing this together with glyceraldehyde, getting this intermediate. And under some conditions, he can make some of these nucleobases. And he's getting really this 5-membered ring, this so-called furanoside. My colleague, Thomas Carell, he looked into a different pathway, slightly more mild conditions. So the trick here is that he's fusing together this precursor for this nucleobase with ribose, but he also needs ribose, which I have to say was probably not available. But then you can fuse it together, and then you're getting also the right nucleoside. It was very interesting. I participated a couple of years ago in a conference in Canada, and I was sitting there, and I had already in mind all the building blocks we get out of our meteorite catalysis. And I thought it's strange that nobody was coming up with the idea, like in organocatalysis, for which the Nobel Prize award was awarded in 2021 to Ben List and Dave McMillan, because nucleobases are amino compounds, and they can activate, for example, here, this would be acetaldehyde. And this can react somehow with glyceraldehyde. And I was thinking about this idea that this might happen really in situ. And I can show you, this is essentially the mechanism of it and how it really works. And it works out also under experimental conditions. So you can take nucleobases, they can activate acetaldehyde, which we obtained from our meteorite catalysis. And now we also know that formaldehyde can react to glyceraldehyde. And here in this reaction, we get a selective reaction, and we get a selective reaction that the stereochemistry, so the handedness of the molecule, is completely controlled, and we are getting out of it these DNA molecules just in a single step. So we have shown this also experimentally, so how these molecules are formed. So this is very interesting. So maybe they can be also discovered probably in space. I'm quite sure for this. And even under a complete mixture, we see already the formation of all these nucleosides in the presence of nucleobases. This is how it works. We also investigated all side reactions. And the question is, so why do we have DNA? And I have to say that the formation of DNA is really absolutely natural. This is not something special, but what we see by this mechanism, and we can assume that this is the real mechanism how DNA also evolved, that this is selectively formed. There is one side product which is also very interesting. I also had to look up the sugar, I have to admit. This is so-called apiose. So from a isomer of glyceraldehyde, so-called dihydroxyacetone, a branch sugar is formed. This is called, we coined the name DAPINA for this. So it's the disoxy apiose nucleoside. And this is quite interesting, but it is not that stable as DNA. And this is just the reason that DNA is formed. Yeah, there were also other proposals which you can find in literature. So also, for example, my colleague Thomas Carell, he proposed maybe the formation of Wöhler RNA. What is Wöhler RNA? You have seen already Wöhler already beginning of 1800 made urea. You can extend urea, so this would be urea, but if you extend it, then you're getting extended molecules, which you can consider somehow as a proto-urea RNA. So if you again attach to this ribose, and we did it. So Thomas Carell is again using ribose just from the shelf, but we do it in situ, and we see also the formation here of this Wöhler RNA. But the question was for us, so has it evolved or didn't it evolve under the conditions of the early Earth? Because there is a problem. We have seen that all these molecules are not very stable. They somehow decompose, they interconvert, and initially it was proposed that they interconvert via this intermediate. Now we are doing a lot of measurements. So we measure kinetic data, we do all these traces over time, look into these molecules, how they evolve, how they decompose, how they interconvert. And what you see here very significantly, so if you start here, for example, from this beta-furanoside, it immediately interconverts into this and then later into this. And so the question was, is this really an intermediate, or and are these Wöhler RNA bases important somehow for the emergence of life? And what we figured out, it's totally different. It's not an interconversion via a common intermediate, but we have interconversion only in this way. So it is like here a square, it always interconverts in one of these directions, but never from here to here. We have only decomposition. So this is the compound X, this is the decomposition product. And of course, we did recently a reaction network analysis. And essentially what came out is that Wöhler RNA is probably or was never involved in the evolution of RNA and DNA, because it is really not stable. So the most stable product is essentially this isomer, which cannot polymerize at all. So this is more or less a dead end. Anyway, so we also looked into peptide formations, for example, also in liquid sulfur dioxide on the early Earth. It's very interesting. SO2 can condense at really ambient conditions. So you need only a few bar of pressure, you get liquid SO2. And we investigated also reactions in SO2. So there was already a proposal that a copper can catalyze the peptide formation, but the problem is that there is only a low abundance of copper, and copper is also not so easily available. But anyway, we did these experiments. It gives a really huge complexity. So we see the formation of all these peptides, and we can follow it up to an octamer, so 8 amino acids fusing together to a peptide. Then the number of combination is so high that it is really hard to elucidate any structure. But we did this, and we were thinking about it. Maybe it is not only copper which is catalyzing this reaction, but maybe also iron. And indeed, we figured out that iron can catalyze this. But the importance here is that we need acetic acid. So only acetic acid can activate iron in a soluble form. And then what happens? This iron acetate, so the salt of acetic acid, can do an exchange reaction with amino acids. It forms a complex. And what we could show, and this is very interesting, there is a so-called effect in chemistry. It's called the Jahn-Teller effect. Yeah, Teller, you know also the name. He was involved in the nuclear program, more or less, so the Manhattan Project. But Teller was also investigating many inorganic complexes, and he discovered an effect which explains essentially here also this reaction. So we get a quadratic square complex where these amino acids are coordinated to it. And then what always happens on the axis here, oxygen is expelled here from the iron. And this is really an important effect which can explain the formation of the peptide bond and the extrusion, so the removal of water from this reaction under this Jahn-Teller effect. Very interesting. And we did this, and we could show that also iron is able to activate amino acids and can make short peptide sequences. Okay, so this sounds very promising, and it is like always in this field that people think, oh, now we are very close to explain maybe the emergence of life. But I have to say there are a number of issues, and maybe I have a solution for this. And the issues are the following. So you have seen that, for example, in this sugar forming reaction, there is no selectivity at all. Yeah, this is the so-called ribose problem. This is really a problem. But I think we have somehow solved this problem. But still, there is another problem. We get random peptide sequences, and we get also random nucleotide sequences. This is not something we want to have. It's not only random, there is also no selective replication. In the case we have a successful sequence. And you can do a very, very simple Gedankenexperiment. If you take only 250 building blocks and you just calculate the number of possibilities you get out of it, then you will realize that if you make only a single molecule of these possibilities, you would need the mass of our Earth to get just a single molecule, a single polymer of these molecules. And then you will realize that this is not something that came out by a probability, but maybe there was another pathway which we do not see, maybe do not yet see. It's kind of like a hidden layer in the system. Okay. So it is also very important to say that molecules alone do not constitute a self-sustaining system. Yeah, this is our experience. Maybe from our breakfast, we have an egg. Yeah, we take this egg. Essentially, all ingredients are there, but you will see that you cannot make life out of it. So this is not so easy. It's exactly this problem. You would have exactly all components together, but it's not so easy to make life out of it. Okay, and now here comes really an important discovery which we made. And this is something that has been, I have to say, just overlooked. So of course, again, going back to our meteorite catalysis, and this is our idea. We want to understand this really from bottom up. We want to understand it, how life emerged really from the formation of our planetary system out of an atmosphere. How did this happen? And so we go again back to our meteorite catalysis. We know from our meteorite catalysis, we got probably on our planet, on our Earth, lakes of aldehydes and ketones. So these are these oxygenated products. And of course, what we can do, and I've shown it already also, which was discovered in 1850, we can do Strecker synthesis. So we can make amino acids. So this was the initial idea. So for this, you need ammonia, you need cyanides, you get an intermediate, this aminonitrile, and normally this is hydrolyzed, and at the end you get amino acids. What is really interesting is if you do this under more realistic conditions, so not under these very clean laboratory conditions, but you do it also in presence of H2S, and we have still a lot of these oxygenated products. And then this was very interesting. So my students did this in the lab, and they came to me and they said, yeah, we analyzed this mixture. It looks like amino acids, but it only looks like something is wrong with the mass. And initially what I thought, because we are doing high resolution mass spectra, that maybe we have only a deviation of a few ppm. But it turned out it's a deviation of 28, 56, and so on. And I said, of course, this is a different compound. And what we figured out, and this is always the difficulty. So if you have to identify a compound which you do not expect, this is really difficult. And this compound here, this cyclic compound, is formed in up to 40% yield. So this is not something you are looking for in some per mil or maybe lower amount in the lower percentage amount or so. But this is 40%. This is formed in high yield. And this compound, it has a little bit of a complicated name. It's a so-called imidazolidine for thione. Yeah, it has here a thio group. And when I saw this compound, I became really excited because what I realized is this is an organocatalyst. So I mentioned it already in 2021. Ben List and Dave McMillan got the Nobel Prize for their discovery, and this here is a prebiotic organocatalyst. Now, what can do this prebiotic organocatalyst? It's very interesting. So here we have somehow a hidden amino acid, very important. So this is a backbone. We can control chirality, so the handedness of molecules with these compounds. We were able to show that it can act as an enantioselective organocatalyst, so making just a single-handedness of molecules. And furthermore, not only this, sulfur can be activated here by light. So here is missing a nu, H nu, and it can be activated and can make radicals. And this is very important because with this, we can utilize now the sunlight to store energy in form of chemical energy. And this is very, very important because to build up more complex molecules, we need energy for this. Here you see the Nobel Prize winners from 2021. So Dave McMillan and Ben List. And as I said, here, Dave McMillan discovered this catalyst. So, and you see now the structural relationship, and we have this catalyst, and our catalyst is working a little bit different. So we don't need a metal because we have here the sulfur. Sulfur can be activated and is doing essentially the same chemistry like the McMillan chemistry. And what is very important, and of course, this came up immediately to my mind. So we have small carbonyl compounds, aldehydes, and so on. And this catalyst is ideal to exactly modify these aldehydes. So it can activate them, introduce here, for example, substituents, and we are getting now modified aldehydes. And this means that this catalyst is able to modify its own building blocks. Another really rare property, it makes enantiopure crystals, so-called, these are crystals which are for conglomerates. So, and this is very important maybe to trigger exactly the single-handedness which we need for the emergence of life. Okay, so we focused on this organocatalysis, and as I said, in the reaction mixture, the catalyst is formed, and this catalyst is modifying its own building blocks and generating now new aldehydes, which can again form a new generation of these kind of catalysts. And not only this, by this organocatalysis, we can make 11 out of the 20 canonical amino acids. So typically on meteorites, you find up to 220, roughly 220 amino acids. But here with this catalyst, we make exactly selectively the amino acids which we know. And these are now 11 out of the 20. So we investigated also this mechanism, how this works. I don't want to go too much into detail, but the mechanism is slightly different. So essentially, sulfur is involved here, activating these molecules and then modifying our initial aldehydes. We have also X-ray structures. We have structural proof for this. We did kinetic studies. And what is maybe very interesting, so there is a parameter which is very important, the so-called nucleophilicity of these compounds. And this is exactly between the catalyst, which was discovered by Dave McMillan and here Jürgensen. So essentially this prebiotic catalyst is exactly filling this very important gap between these two catalysts. Okay, there is another surprising property. These catalysts make not only their modified building blocks, but they can do also an exchange reaction. And the exchange reaction can happen here, so forming here a second generation catalyst with any carbonyl compound, or as I already pointed out, this catalyst is catalyzing the modification of its building blocks and making now a totally new type of catalyst. And we investigated this in detail. So we started with the pool of aldehydes and ketones, which we obtained from meteorite catalysis. And what we saw over time is the formation of a first generation, second generation, and third generation catalysts. And it's very interesting. They have partially different properties, and also depending on the environment. So if there is more ammonia or just pure water, for example, we enrich certain catalysts, and they catalyze also different reactions. And this is somehow related to a Darwinian evolution on the molecular level. So to show you that these catalysts not only catalyze their self-modification, but they do also other reactions. They can, for example, also activate phosphates. And this was, I would say, a problem for a long, long time, because what we know from our world of biomolecules is that we need these nucleotides, which are phosphorylated in this position. This is the 5 position. So it's a 2, 3, 4, 5 position. This is position 5. And this is really hard to explain. So all other people in the field, when they do experiments, they have always the trouble that they get phosphorylation in the 2 or 3 position. But for polymerization, normally 3 and 5 are good, but 5 is better to achieve exactly the formation of these polymers. And this is what our catalyst is doing. It is exclusively doing the phosphorylation in exactly this position. And this is something what happens also then in solution. So you have now your nucleosides, they get phosphorylated in the right position. They can be activated again with the catalyst, and now come in these dark and night cycle, night and day cycles. So the dark and the light, which is activating essentially this compound. And when it is activated, we are getting here a photochemically induced oligomerization of DNA and RNA. So here, only to show you that it is also possible, for example, to fuse together a DNA building block, so where the OH group is missing, and RNA building blocks, so they can be fused together and can be oligomerized. We were thinking about it, and now combining somehow our DNA synthesis with this, where we have shown that we get selective formation of ribose with this activation chemistry, with this organocatalyst. And a very, very important prebiotic cofactor, which is also important for us. So we are making, let me say, every day in our body, several kilograms of this compound over time. So it's always at low concentration, but if you would sum it up, you make several kilograms, because NAD+ is a very important hydrogen transfer reagent in our body, very important to reduce compounds. So this has always something to do with the transport of molecules and the energy in our body. And we looked into it, and we figured out, yeah, we can start here just with ammonia, and we get here this amino ribose selectively. And the only step which was proposed by Orgel here in the Orgel type NAD+ synthesis, so he proposed here a pathway to this building block, but he figured out that essentially only this step here is working. So we can take this step, which makes sense, fuse it into our reaction, and then we are getting indeed this building block, which was intended to be synthesized by Orgel, but he never achieved it. And then we can move on. So water hydrolyzes it. So we are getting here an amide. We see activation again with our organocatalyst. We can phosphorylate it in the right position. And now we fuse it together with an activated adenosine, and we are getting at the end NAD+. And it was very interesting. So a colleague working in this field of cofactors, he contacted me and he said he was totally surprised. He didn't expect that NAD+ probably evolved so early in this reaction network on our early Earth. So he expected that essentially this cofactor was developed maybe later in the evolution or evolutionary process, leading finally to the biochemical machinery. But this is, I think, also very, very important. So we have already NAD+ in hands to mediate, for example, metabolites in cells. Okay, and then we made another exciting discovery. So I always ask my students, just do a very, very simple reaction. It is the so-called aldol reaction, which is more or less the basis also to make sugars, for example. And this is fusing together just two carbonyl compounds like aldehydes, which we obtain from meteorite catalysis. And then one of my students came to me and said, it's strange, I can isolate the product. So normally you expect that A and B reacts to a product C, and you want to isolate it. But he couldn't isolate it because he said the solution is getting always very milky after 2 hours, and then I get a mixture which I cannot isolate anymore. And I said, okay, this is maybe interesting, so just go under the microscope. Look what happens on the microscope. And what we saw is, and initially we thought we have a bacterial contamination in our lab, we saw the formation of cells in this reaction. So starting from acetaldehyde, what happens there is it gets oligomerized to lipid type molecules, and they automatically assemble into a protocell. And I want to show you this here. This is one of the first videos we obtained. So this is a video over 2 hours. I condensed it to some minutes. And initially you see almost nothing, but then you see there are some dark spots here in the solution, like droplets, kind of a droplet formation. So this is initially what we expected, but I would like to accelerate this a little bit, maybe, if it is possible. Oops, no, I go back here. I would like to, oops, now I somehow, here. I would like to accelerate this maybe a little bit in time. Now let me see how I can do this, because normally it shows up. Yeah. Now we have to wait until it's going through. But you will see, you see this is getting more and more crowded by these droplets. And you see more and more bigger structures. So they get into the focus and out of the focus here under the microscope. And this is very, very interesting. So after 2 hours, it is completely full of these kind of cells. So initially, as I said, we initially expected that these are only droplets, but then we did more careful analysis. So this is more or less after maybe 1.5 hours. This is how it looks like. And as I already pointed out, we thought this is a bacterial contamination, but I would like to stop it here. So we can follow up this also by high resolution mass spec. And it turns out that acetaldehyde oligomerizes to lipids of a length of, as you can see here, of 1820 approximately. So the average at the end is around 18. So this is also what we see in normal cells. So we have the fatty acids, C18 is very, very common. So we can show this, and we elucidated also the mechanism, which is a little bit complicated because there are several catalytic cycles and they go hand in hand. So sorry for maybe overloading you with the chemistry, but what is really exciting at the end, the outcome is we have unsaturated, partially unsaturated oligomers of lipid type molecules. And not only this, what we figured out is that our catalyst can do again the same exchange reaction which I've shown you in this evolutionary catalytic cycle, and the catalyst gets attached inside these protocells. And this is the reason that we see a kind of an autocatalytic effect. And what is really interesting, in the first phase, kind of droplets are formed, and then water is formed in this reaction. It is expelled from the cell, and at the end we have here a protocell with a double layer after 2 hours. And this is very interesting, and they are catalytically active. And now you can imagine what our next steps are. So we are looking into reactions to drive, for example, a metabolism inside these cells. And very important, they overcome a concentration problem, because what we already see is that these cells are able to concentrate the organic material inside. So I have to say I'm quite optimistic that maybe in the next couple of years we see maybe a kind of chemical life in these systems. I want to show you something different. This is a little bit an outlook where we are at the moment. We are looking also into processes because, as I pointed out, if we have amines, these amines can selectively form ribose here at this position. And the same happens with our catalyst. And it is, I think it's almost clear that this catalyst is making also a 5-letter code. So a code we know from RNA and DNA. And this is maybe a true progenitor of RNA and DNA because it can form this code. And now you can imagine that we can make oligomers out of it. And what is very interesting, because we have here the same patterns in these catalysts, like in RNA and DNA, they can form interaction complexes, they can form complementary sequences, and this means we have a very simple mechanism to copy them. So we can oligomerize them again, we copy them, and then at the end, and this is always the big question, because normally for the translation to function, we need a ribozyme to translate it into a peptide sequence, because these are not catalytically active. But now remember again, what I told you is that essentially here the backbone is a hidden amino acid. When they hydrolyze, and they can hydrolyze, and they oligomerize, we get in a reproducible fashion peptides. And this is very interesting. This is where we are. We are working on this. So this is an outlook. This would be a ribozyme independent system, which is able to store information, successful information, can copy this information, translate it, and give function. And with this, I would like to summarize. So I've shown you, maybe it was really a long trip, but our life didn't emerge maybe in a few days, but in a couple of years or hundreds or thousands of years and so on. But we started here with the meteorite catalysis. I've shown you that we can form very simple building blocks. These very simple building blocks can form an evolutionary catalytic system. And this is very interesting. I didn't mention it so far. This catalytic system shows Darwinian evolution, and to interpret in a modern fashion Darwinian evolution, this is kinetic evolution. So this is also what we know from daily life. So the fastest system will always overrule the other systems. And this is, I call it maybe in a modern fashion, it's a kinetic selection. So the most active catalyst will be the catalyst which dominates at the end the system and is catalyzing other systems. And as I've shown you, we are really about to get true function in these systems. And I think it's now really exciting that we see already the formation of the first protocells. And now I'm optimistic that maybe in the next 5 to 10 years, we will really see somehow a self-sustainable chemical system which can represent kind of chemical life. With this, I would like to thank first my group members who worked in this field. I would like to thank also the funding agencies, Volkswagen Foundation, DFG, of course, also the Origins cluster. So we have now again a second phase here in Munich, which is great. Our collaborative research program about origins of life, and also Max Planck Society. And of course, you for your very kind attention. Now I'm looking forward to the discussion and questions. Thank you so much.
Yeah, thank you, Oliver, for such an in-depth and very comprehensive seminar.
Okay.
We don't have like much time, but if anybody has like any questions that they want to ask, or you can also write in the chat.
Mm-hmm. Maybe I can look into it. Yeah, there are a number number of questions, I think.
Well, yeah, thank you very
Steve,
much, Oliver.
hello. Nice to see
Great.
you. Yeah.
Great to see you too. As I wrote in the chat,
Yeah.
very, very comprehensive. I really liked how you began
Mm.
with the historical
Yeah.
basis. I wanted to add the name of Walter Loeb, who's often overlooked,
Okay.
and this generated a little bit of discussion in there. And then, you know,
Oh.
the liquid sulfur dioxide is interesting because many volcanologists well, not many, a few have proposed that you can stabilize liquid SO2 just below the surface in basaltic systems that
Yes.
subsequently degasses as SO2. So I've put a little bit in
Yeah.
the
And
chat box there. So it's more like commentary rather than like I have any specific question at this time.
it's very interesting because what most people overlook is so very often you think SO2 is only reacting, for example, with water, of course, making
Yeah.
sulfides and so on. Sulfides are important, for example, also to stabilize aldehydes because they can form these complexes. But it's also interesting that SO2 as a liquid has some really mild activation function. So it can mildly activate molecules, and it's very interesting. So even industry is looking into
Yes. Yes.
at large scale because it's very nice. At the end, you remove it; it just removes, and it is able to stabilize certain salts and things like this. And this could be very, very interesting. Yeah. Yeah, but it's a very, very good point. Yeah, I just I can add also Loeb to this, which is very interesting because he looked into these amides. If you get dehydration, you end up again in this nitrile chemistry, which is also
Yes.
interesting. It's a
Well,
good point.
thanks again, Oliver. I
Yeah,
appreciate it.
thank
Look
you.
forward
Thank you
to
for your
further
comments. Yeah.
discussions.
Yeah. I think what is really important here is to see that a catalytic system evolves really beyond, let me say, the biomolecules, because we always try to just reproduce this. But the problem is how to get there. And this is something you can really bridge with these organocatalysts. And these organocatalysts, as I said, they are formed in up to 40% yield. This is not something which is very rare, but it has been just overlooked. It's very interesting. Okay. Let me see if there are more questions.
Hi, Oliver. So
Yes.
I mean, the big question was initially, as you well know, first to form, you know, this key like building blocks. Now it's kind of seems to be solved,
Yeah.
and then the next question is the condensation. You know, the making the chain,
Right.
and it looks like you started tackling this. It's really interesting. So do these things work also in the presence of water, or you need these lipids to isolate them from water to accelerate the condensation reactions? Or
Yeah, it's a very interesting point. So luckily, or I would say we need the water that this oligomerization stops at C18. So it's very interesting. Nobody is asking this question. So if you take a textbook, for example, biochemistry, and you read it, and then this is really funny. It tells you that yeah, there is a kind of aldol reaction to form these building blocks, and then the comment is always this happens 6 times, and then you have C18. So it seems that people think that it is programmed. It is somehow programmed maybe in DNA or then translated into RNA, mRNA, and so on. But this is just not true. The thing is, you have a phase transition. So this is, in this sense, somehow physics or physical organic chemistry. which gives exactly this border. And this is, for us, I really have to say, we were amazed about it, that it doesn't polymerize maybe completely, so you would have like a organic polymer, but that it really stops there, and then it makes these cells. And that's really amazing. Yeah, amazing structure. And I think it is maybe really important to explain that we get an enrichment of the organic molecules inside the cells, because this is the next problem. So when people discuss, oh, we do the chemistry, we have a pond of molecules, yeah, maybe there are wet and drying cycles, lagoons like this. But again, then we are in this trouble. These are very, very special reaction conditions. But here, what we see, maybe life is more robust. It just evolves, and in this sense, I'm really excited about it, I have to say. And also maybe Dima, what you also say. So we are thinking at the moment a lot about the nitrogen activation, because this is also overlooked by most people. Most people say maybe it happens at the same time, but they do not have this catalytic background. And if you understand this process, then you realize that nitrogen was activated after the sky was cleared from CO2. And this is very, very important, because otherwise CO2 kills more or less all processes. So industry knows this. Yeah, so that's a real challenge. Yeah.
Thank
But
you, Oliver.
fantastic.
Yeah, very interesting. This was
Yeah,
very
very.
interesting.
It's super interesting. Yeah. Okay. Let me see. Yeah. So Stephen was addressing also the oligomerization of RNA and DNA monomers. So we are working on this, and it's also interesting now. It turned out that one of the side reactions of our catalyst is that we see the formation of nucleosides which are connected via phosphate. And initially we thought this is maybe not a good step. Yeah, it's like a side product. Now, recently I met a former coworker of Czech Šostek, Daniel Dušević, and he told me, oh, this is really fantastic. You have the building blocks we are looking for, because this is an activated nucleoside. And now we will really challenge this question: can we get beyond the current limit? And the current limit is approximately 19 nucleosides to get an oligomer. If we really get from bottom up beyond this limit, and this would be fantastic. So if we manage this, then yeah, maybe it's the next step. I'm always saying we are the observer somehow. We are observing. We are scientists. We have to observe what happens. It's not that we are really directing it. We can help it to accelerate the whole process, but we have to observe, and we have to observe very carefully and try to understand what happens and what are the implications. That's important for me. Okay. Further questions?
It does not seem so.
Okay.
I want to thank you again, Oliver, for coming and giving us a really great talk.
Yeah, thank you for having me here. Yeah, wonderful. Thank you so much, and have a nice day. Yeah.
Yeah, have a nice day.
Thank you.
Bye.
Bye. Super. Okay, wonderful. Okay, then see you. Thanks.